Thanks very much. It's a pleasure to be back here and in the design school. I'm going to take the liberty of talking a bit about something that's more biological than engineering based in terms of some of the things you think about all the time, and talk about things that have been engineering themselves for hundreds of millions of years and are some of the most elegant little killing machines we'll find on the planet. They were finally being able to bring into medicine to serve as therapeutic tools to treat multidrug resistant bacterial infections, namely bacteriophages. To get a sense of where we're going with drug resistance, it's increasingly clear that multidrug resistant bacterial infections, ones that we can either not treat at all with antibiotics or treat with only the most toxic antibiotics, are spreading around the world and causing an increasing number of deaths. In 2019, there were about 5 million people who died with antimicrobial resistant organisms, and that's projected to continue to rise dramatically through 2050. When you look around the world, this is not just a problem in places where there are a lot of fancy antibiotics. It's actually present. It's a problem around the world. Including, in particular, Latin America and the former Soviet Union, as well as the Middle East. And when one looks at what we thought was going to kind of continue forever, the development of new antibiotics, what's becoming increasingly clear is that we're running into a situation where our antibiotic pipeline is running dry. If you look across the top of this bar, you can see the year in which each of these new classes of antibiotics was introduced into clinical medicine. Below that, you can see when the first clinical isolates were obtained from patients that were no longer sensitive to these antibiotics. And you can see almost reliably every time an antibiotic came into existence within a few years, new organisms were evolving in patients and in our environment that could no longer be killed by these antibiotics. Right now, they're really, when you look at the antibiotic pipeline, it's really quite dry, despite the increasing number of people who die each year of these multidrug resistant infections. So against this backdrop, I'm going to tell you a little bit about a story of a patient that I encountered in 2015, Tom Patterson. Tom is a faculty member here at UC San Diego, a member of the Department of Psychiatry. His wife, Stephanie Strathty, is a professor of global health, and Tom and Stephanie went on a vacation to Egypt in November of 2015. And while they were floating up the Nile on a barge, Tom developed excruciating abdominal pain and began to have a fever. Stephanie got in touch with me and asked what she should do. We talked a little bit about what was going on, and I told her I was concerned that he had pancreatitis and that she should get him to a hospital relatively quickly. Tom, at the time, was in his mid-60s, was a little bit overweight, and had diabetes, and really didn't need to be in this state where there was not a lot of medical help around. Now, normally we don't talk about patients by name. In this situation, Tom and Stephanie have written about their experience in a book, The Perfect Predator. So those of you who want to call the HIPAA police, feel free. The Perfect Predator tells this story in much greater detail. But to do the story at 35,000 feet, it turned out that Tom did have pancreatitis. When he got to a hospital in Luxor, he had a pancreatic pseudocyst. You can see that little green cross here. That's a bubble that shouldn't be there, and it's filled with pus behind his stomach. That was later found to be filled with an organism called Acinetobacter baumannii, which is an organism that is highly resistant to antibiotics and is able to pick up antibiotic resistance very easily from other organisms. It's been a major problem for our troops who were in the Middle East at the time, and is a real problem throughout the Middle East. Tom was stabilized in Luxor, flown to Frankfurt, where he was given antibiotics, and they punctured that cyst through his gastric cavity and grew this multidrug-resistant Acinetobacter. He, after about a week in Frankfurt, was air-evac'd to UC San Diego, arrived here just before Christmas, and spent about 3 months over at Jacobs Medical Center getting increasingly ill, so that by the time March rolled around, he was confused, he was intubated, he was on 3 medications to keep his blood pressure up, his kidneys and his liver were failing, and the pulmonologists, the people who were taking care of him in the intensive care unit, were talking to Stephanie about whether or not they should try to withdraw care and let Tom pass on. At that point, Stephanie saw this paper that had appeared in a microbiology journal about bacteriophages that were able to kill Acinetobacter baumannii. This is not a paper about using them to treat a patient, but rather a laboratory experiment in which these bacteriophages, and I'll tell you in a minute what they are, were found to kill Acinetobacter in a laboratory experiment. Now, bacteriophages are literally eaters of bacteria, and they were discovered about 110 years ago now in the Indian subcontinent when people found that you could take bacteria that grow in rivers there and grow those bacteria on an agar plate, and then take water that is obtained from those same rivers, filter it so that what gets through the filter doesn't include any bacteria, but rather things smaller than bacteria. When you pour that water onto the plate, holes appear in the bacterial growth. In other words, something is eating those bacteria. These were talked about for about 10 years and found in the mid-20s to actually be viruses. In those days, we didn't know much about viruses. Influenza, for example, the great influenza that killed tens of millions of people around the world. It wasn't until 1929 that we realized that that was actually a virus that killed everybody around World War I. So these were thought to be some people thought they were proteins, some people thought they were toxins, but it took a long time to realize they were actually viruses that were able to attack and kill bacteria. Now, the basic life cycle of a phage is relatively simple. If you just focus on the outer circle here, the little predator-like orange structure on about 3 o'clock there on the right is landing on the surface of a bacterium and injecting its DNA into the inner part of the bacterium. That DNA then makes proteins as we go down beyond at 6 o'clock and 7 o'clock of the phage, and replicates the phages 100 to 1,000 times within that bacterium over the course of about 20 minutes. That bacterium is then blown up by lysins, which are another set of proteins that the phages make to blow up the bacterial cell wall, to release them to go on and attack other bacteria. So these are really very efficient bacterial killing machines. We'll talk about the inner circle in a few minutes. Now, for a while, people were very excited these might be useful to treat bacterial infections. And indeed, in the 1920s, they were marketed in the US and Europe and in the former Soviet Union as treatments for infections. They were named after the type of disease you had. So if you had, for example, a nose infection, the product was called a bacteriophage. If you had diarrhea, it was called Bacteriophage. Now, the problem with this was that although there was an effort to grow phages from the places that these clinical syndromes were occurring, no one understood the complexity of the interaction between individual phages and individual bacteria. So these were mixtures of phages that were, each batch of phages was different, so that one batch of a Bacteriophage might include 15 different types of phages, and the next batch that you bought from the same person would have 15 different ones. So it was a very poorly controlled sort of experiment, and very little was done in terms of rigorous medical research to understand how to use them. There was so much excitement about them, though. If you remember the book Aerosmith by Sinclair Lewis, that book was actually about bacteriophages and the promise they held for treating infections. It's a very interesting book about an emerging technology at a time when the technology was not completely understood. So phage therapy fell out of favor in the Western world when antibiotics came along. And It was in part because the research that was being done to try to understand how to use them wasn't that rigorous, but also because antibiotics were so easy. You can make a molecule, you know what it is, you can make it again, it's the same molecule, and you can develop antibiotics that will kill a wide range of different bacterial species. And so from the standpoint of a physician who wants to treat a patient with an infection, it's great to have an antibiotic that will kill a lot of different things. If you're a commercial company and you want to sell something, if you have something that will kill a lot of things, that's better than having something that will kill only one thing. So before too long, antibiotics replaced phages completely in the US, Europe, Australia, and the Western world. They continue to be used, however, in Russia, Poland, Georgia, not our Georgia, but their Georgia. And they were used for treating infections because their antibiotics were expensive and phages were in use. And this paper that I told you about had come from Georgia, a lab there that had been working on phages for about 70 years. So when Stephanie saw that paper, she called me and said, would it be possible to treat Tom with bacteriophages? And I said, well, that's kind of an interesting idea. I can't really say with any great degree of certainty that we'll be able to treat him successfully, but I can tell you we're not really making much progress with what we've been doing the last 3 or 4 months, and I'm willing to try to see if we can at least pursue it for a while and see what comes up. The questions, though, that were in front of us were whether we could find phages that would be active against Tom's specific organism. One of the problems with phages is also one of their virtues. They have a very narrow spectrum. An individual phage will only kill a certain percentage of bacteria of a specific species. So if you have a patient with an Acinetobacter, you have to find a phage that will kill not only just Acinetobacter, but that Acinetobacter. And that can be quite difficult. The next question was, if we found them, could we make them in a great enough quantity and at a great enough purity to actually give them to him safely? And how would we even give them? They haven't been used in the West for about 70 years. And when we used to use them, we used them by pouring them on wounds, having people drink them for diarrhea, having people inhale them for nasal infections, but they weren't used like antibiotics intravenously. So luckily, we were able to find some phages through some serendipity. The paper that I showed you earlier from Georgia, one of the co-authors was a physician named Jean-Paul Pernet, who was from Belgium. Jean-Paul works at the Royal Military Hospital in Belgium and was using these phages to treat burns in soldiers. Stephanie Strathey got in touch with him, and he said, I'd be glad to send you those phages, but they're already in Texas. I collaborate with a guy named Ry Young, who's at Texas A&M. Why don't you call Ry? So Stephanie got in touch with Ry, and Ry said, well, good grief, it's always been kind of interesting about treating patients, but you know, I've always been worried about that. It might kill him. And Stephanie said, well, Tom's dying. I'd really like to try them on my husband. And Ry resisted for about 3 hours, and finally it was Friday night. He said, oh, hell, I've got a graduate student who's not this busy this weekend. Send us the organism, we'll see what we can do. So we sent the organism down to Ry, and within about a week, he found 4 phages that were active against Tom's organism. It didn't happen to be that the Georgian phage was active, but he had some others that were active. In the meantime, I'd gotten in touch with the FDA, and they told me that there were people at the Biomedical Defense Research Command of the U.S. Navy in Bethesda working on phages for Acinetobacter for treating our soldiers. And I got in touch with Theron Hamilton there, who was a commander in the Navy, and he said, well, send me the organism. We'll see if we can come up with phages that are active against his organism. We had 4 more phages from him. Now, this shows you how we know the phages are active. If you look at, this is essentially a respirometer that measures on the y-axis respirations of bacteria. And you can see in the black line when there is no phage present, bacteria, in this case Acinetobacter from Tom, grows relatively quickly. But you can see when phages are added with each of those colored lines, either by themselves or in combinations, the organism doesn't grow. It suppresses growth. These phages suppress growth. So we found phages that were active. Then we had to get approval from the FDA, and it turns out getting approval from the FDA for something like this is very easy. I was concerned that it was going to take us weeks to get them to approve it. But when you have a patient who's an extremist like this, about all you really have to do is get on the phone, call the FDA, tell them what the story about the patient and why you would like to treat the patient with what you would like to use. If it makes sense to them and the patient consents, they can give you approval over the phone. And I was lucky enough to get in touch with a woman named Cara Fiore, who was a very smart bacteriologist who had been who was regulating fecal transplants and was bored to tears doing that and said, I've always wanted to do something more interesting. This sounds great. Let's do this. So the regulators really became a major help in moving this forward. We then had to decide what dose to give and how to give it. We knew that with where we were growing Tom's organism, by this time from his lungs, from his peritoneal cavity, from his urine, sometimes from his blood, that getting him to drink the phages wouldn't get them where we needed to go, and we needed to give them intravenously. And we also knew that we wanted to give him as many phages as we could because he had such a widely disseminated infection. The way we found out the way we determined the dose was to look at the purity of the phage preparations we were able to make. When you make phages for clinical use or for the laboratory, you basically take a phage you want to amplify, pop it into a flask that has bacteria that will grow the phage, that the phage will attack, let the phage attack that flask of bacteria, and then you have a bunch of dead bacteria and then phage floating around in the supernatant fluid. You then filter that supernatant fluid through a filter that keeps the bacteria back but lets the phages go through. But then you have a bunch of endotoxin, which is a product of bacteria that is left over when they're killed by antibiotics or they die or phages kill them, that's quite toxic for people. And so the next step is to try to purify the phage preparation to reduce the endotoxin. And we were able to measure how much endotoxin was present, and we knew how much endotoxin was safe to give a patient, so we back calculated how much phage we could give Tom based on the purity of the phage preparation, and we were able to calculate we could give him about 10 to the 9th active phages every hour. So we began to do this. On the left is Andrew Peisel, who was one of the interventional radiologists. We first gave the phages into his abscess cavities. He had a bunch of abscesses in his stomach, and we had catheters that were draining the abscesses, and what Andrew did was instilled phages into those catheters, wash them around, and then let the phages stay in the cavities. We started doing that on a Tuesday afternoon, and by Thursday he was not much better, but no worse. He'd been kind of doing this all along, but he was at least stable. And at that point, the first phages we used were the ones from Texas A&M, and by the 17th, phages came from the Navy. We decided it was necessary to treat him parenterally, in other words, intravenously, to get the phages throughout his body. And we gave him that same dose of phages from this time the Navy intravenously. And over about a 48-hour period, he stabilized and woke up. His blood pressure medicine, medicine support we were able to decrease that. His kidney function improved. And before long, he was extubated, and after some complications of just being older and in the hospital, he got out of the hospital and went home. Now, one thing we saw over the course of his infection is that, of the treatment, is that his bacteria became resistant to the phages we were using. And you would expect that because one of the things that bacteria do when you treat them with antibiotics or with phages or anything that kill them is that the part of the population that's less sensitive to what you're using, as Darwin would say, has a selective advantage, and those overgrow the bacteria that are killed, and you get, in this case, bacteria resistant to the phages. If you look at the upper right corner, that's the graphic I've already shown you, and you can see that in panel F, just below that, another isolate from Tom 6 days later, the bacteria were beginning to grow through the phages. They were no longer being held down for 20 hours in the laboratory. And by day 10, none of the phages were able to suppress his bacterial infection. Now, we were, by this time, though, he was much better, but one of the things that this told us is that we were doing something that the bacteria didn't like, and they were having to, they were being selected to no longer be as sensitive to the phages. Now, this issue of phage susceptibility has been going on, of bacteria has been going on again for 300 million years. Phages attack bacteria. Bacteria evolved to try to evade the phages, and they have elegant mechanisms to do that. I'm not going through this entire graphic. It's a great review if you want to read more detail, but those of you who think that, for example, endonucleases were created to allow us to move genes around, not the case. Endonucleases were things that bacteria developed to evade phages. Those of you who think that CRISPR-Cas was developed to give Jennifer Doudna the Nobel Prize are wrong. Phages push those into bacteria. Bacteria develop those to chop up phages on the way in. So a lot of the tools we have in molecular biology now were things the bacteria themselves developed to protect themselves from phages. Phages, on the other hand, turned around and tried to resist that. They methylated their DNA so that they weren't as susceptible to some of the restriction enzymes. Bacteria upped the ante and developed restriction enzymes that would chop up methylated DNA. So this evolution has been going on forever and ever. In Tom's case, we were able, though, to take that resistant bacteria from day 10 and go back to the laboratory and find new phages that were active against his bacteria that had become resistant to the first wave of phages. So one of the nice things about phage therapy is that when resistance developed, you can go back in with another round of phages that are active against the new bacteria. And when we did that, you can see in blue over, this is a dendrogram of phage DNA. The red circle are the phages around the phages that we first used. You can see they're genetically quite similar. But the new phage, this protophage in blue, is the one that we found that was able to take out his day 10 organism and gave a new round of treatment for his disease. Now, what did the bacteria give up to be able to grow in the presence of those original phages, the red ones? It turned out what the bacteria gave up was its ability to secrete a capsule. If you look on the left, you can see this big juicy capsule around each of these bacteria, and you can see that that juicy capsule is less visible on TP3. And the phages that were attacking his bacteria were latching onto that capsule as their way into the bacteria. So the bacteria said, okay, I'll just drop the capsule, but keep growing. Now the problem for the bacteria when it does that is it's much more susceptible to being eaten by macrophages and by polymorphonuclear cells. So although the phages were pushing the bacteria to drop the capsule, the bacteria were basically dropping their defenses against Tom's polymorphonuclear cells and allowing his immune response to help out in terms of curing the infection. So there are a lot of benefits to phage therapy that aren't just directly related to killing the organism itself. Now this is Tom over in the Student Support Center, standing on the balcony there about a year and a half later, or 3 years later, actually, I'm sorry, with Theron Hamilton when he came to visit. And Tom had lost about 100 pounds. He'll tell you this is not the way to lose 100 pounds, but he's actually doing extremely well now and walks about 3 miles a day and has become a spokesperson for Phage Therapeutics. Now, what has happened since that? Well, we were impressed by that case and had another couple of cases we got involved in and thought we should try to see if we could pursue this and have a more organized approach to be able to help more people. So we put together a center here at UC San Diego called IPATH. It's a center that focuses on the uses of phages for medical use. We have two goals. One is to be able to provide phages to people like Tom who don't have other options, and the other is to develop a rigorous phage research program. To get to the phage treatment part of it first, we have a coordinator who gets calls from patients and physicians and patients' families, and we then evaluate with the patient's physician whether or not a phage is the right approach for them. Most of these requests come from outside UCSD, and if we think a phage is something that would help them, we then try to start working on going after phages that could be used in their case. To give you a sense of the scale, we've had about not quite 2,000 requests over the last 4 years for help with phage therapy, but about 3/4 of them turn out to be people who either don't have a bacterial infection or that we can help by helping the physician change the antibiotics around. But once we start looking for phages, we can find an active phage in about half the time before either the patient gets better or is no longer needing a phage. And when we treat patients, we're getting about a 50 to 60% success rate. Now, where do we find the phages? We have a consortium of collaborators at different universities that each one of which works on a specific bacterial species. So, for example, Texas A&M, they work on Acinetobacter. At Walter Reed, they work on Pseudomonas. Here at UC San Diego, we work on Enterococcus. And so depending on what organism a patient has, when someone calls us and we decide a phage would be useful, we then arrange to send their organism to a lab that has phages for that particular bacterium to initiate a phage search and get care there. Since that time, this is Saima Aslam in the upper panel and Darcy Wooten below her. These two women have treated, particularly Saima, patients here at UC San Diego. Saima wrote up her first 10 cases in a journal and published them about 2 years ago. And when you look in the literature, you can see that case reports of phages, phage therapy in the medical literature have really picked up since Tom's case in 2015. When you look and see what people are being treated for, it includes patients with implanted devices. Now, these would be infected hips, infected joints, prosthetic joints, pacemakers, Dacron aortic grafts. And the reason these are very difficult to treat with antibiotics is that bacteria put up biofilms on the surface of foreign bodies and they can grow in these biofilms very slowly, and it's quite difficult for antibiotics to kill them. Phages disrupt biofilms, so that one of the uses of phage therapy has been to disrupt biofilms and allow us to sterilize prostheses that otherwise would have to be removed, have the wound sterilized, and then a new one put in. So this is one of the areas that we're quite excited about. Pneumonia, osteomyelitis, bone infections are other areas that phage therapy has been used. So these are some of the areas that we're pursuing. I'll talk to you a little bit in a minute about mycobacteria. Tuberculosis is a mycobacterium, and there are cousins of TB that cause particular problems in immunocompromised patients that we've been using phages for. So what are the bottlenecks to going further? Well, we obviously need to develop a broader spectrum of phages so that it's not as hard to find them when a patient presents. And we need to improve phages to have them have a broader host range, to be able to kill better, and to be able to disrupt biofilms better. And we now know how to engineer phage to do this, and that's underway in a number of laboratories as we speak. And what about engineering phages? Again, I'm not going to go through all the details of this. Again, from a nice review, this is one that Stephanie Strathty and I and a couple of colleagues published in Cell earlier this year. But there are multiple ways that you can swap out DNA segments of phage in a directed way to help the phages either have receptors that are able to go after a broader range of bacteria or have a better means of killing bacteria once they are in place. And these are underway in academic laboratories and in biotech companies around the world. Now, can engineered phages be used to treat patients? Well, the first patient with an engineered phage to be treated was a patient that we encountered in the UK, working with Graham Hatfull and Helen Spencer, a very bright infectious disease transplant physician in England. The patient was a young woman who at the time was 17, and she had cystic fibrosis and had developed an infection with an organism called Mycobacterium abscessus. M. abscessus is a cousin of Mycobacterium tuberculosis. We don't have good antibiotics for it. We have some that can work if you have an immune response that helps, but they're not very good in people whose immune responses aren't working, and they're toxic and difficult to take. This young woman had developed an M. abscessus infection in her cystic fibrotic lungs when she was 9, and by the time she was 17, she needed a lung transplant. She did well for about 2 months, and then you can see these lesions on her forearm, and then in the lower left panel along this surgical incision. These are lesions that are just chock full of Mycobacterium abscessus that returned when she was put on immunosuppression to keep her lungs from being rejected. And on the right, you can see that's a PET scan that lights up hot lymph nodes. These are areas where the M. abscessus were growing as well. That's when I got in touch with Graham Hatfield. Graham is at the University of Pittsburgh, and he runs a program called C-phages. We have a program like that here on UC San Diego's campus that Joe Poliano runs. And what C-phages is a program in which undergraduates look for phages that are active against Mycobacterium schmegmatis, which is, again, a member of the same family, but harmless. It's one that you can grow on the top of your tabletop, or you can grow it on your bread or in your wine or whatever, it can't hurt you. So it's used in undergraduate laboratories as a way to introduce people to biology and to phages. And Graham has been running this program for about 15 years and has 15,000 sequenced phages in his freezer that will attack Mycobacterium schmegmatis. He found that some of these phages would actually kill Mycobacterium abscessus. And so I got in touch with Graham, and we heard about this patient. And we thought it might be useful to try to see if we could treat the patient that I showed you. Now, the problem with mycobacterial phages, we're getting back to our life cycle, and this time focusing on the inner circle, is that mycobacterial phages are ones that are what we call lysogenic. They instead of turning on and replicating themselves as in the outer circle and lysing the bacterium, they go into the bacterium, insert their DNA into the bacterial DNA, and live there silently, replicating with the bacterium, but they don't kill it. Every once in a while, the phage can be induced, and that is become active again, and then go out of that bacterium and attack others, but it's not a very good way to kill a bacterium if you're trying to treat a patient. So what Graham figured out was that you could take out the repressor gene or the integrase gene or both, and phages that would normally be lysogenic no longer are able to be lysogenic, they become lytic. And so Graham developed a technique called bacteriophage recombinering, in which he electroporates a segment of DNA that he wants to remove, co-electroporates it into a permissive mycobacterium, and then selects for colonies that have shorter DNA, indicating a resection of that segment that he's targeted with the oligo. And when he recovers and purifies those plaques, they are now lytic phages and no longer lysogenic. So he created some lytic phages that were able to kill Isabel's Mycobacterium abscessus and use them, and we were able to use them to treat her. You can see here on the upper left is a picture you've already seen. Upper right, these lesions are beginning to go away. Same thing along her incision line. This shows you her PET scan improving. The big black blob down is her bladder, but you can see that where the dye is excreted, but you can see the lymph nodes are quieting down. And if you look at her pulmonary function test, her ability to blow out air in 1 second, you can see that she got her transplant, improved to about 60% of what one would expect her to do with her size lungs. Then when the phage came along, she got up another 20-25%, so that you can see that her lung function improved as well. And she lived now for another couple of years after that, and then ran into problems with graft versus host disease and ultimately succumbed to that, but the Mycobacterium abscessus was controlled. Constance Benson, a colleague of mine here at UC San Diego, has now worked with Graham, and they've treated about 50 patients. They published 20 of them in Clinical Infectious Diseases last year, and are beginning to understand better and better how to use this approach. So where do we need to go further? In addition to this, we need really rigorous clinical research. And the most important thing is it doesn't need to be anything we don't know how to do already. If you think about phages, all they are living antibiotics. They're not anything different from streptomycin or penicillin, and we need to evaluate them the same way. We need to look at patients who have infections with bacteria that the phages will kill, try to figure out what the best dose is, and then look to see how we can help the patient most clinically. That's how we develop antibiotics, and that was what was not being done back in the 1930s and 40s with phages, and so that's where we're returning now. Phage clinical trials are picking up as well. You can see how things have picked up since 2015. Even more so this year. And where we need to go is kind of an iterative process in which we do clinical trials on the right and study PK/PD, which means it's a shorthand for knowing how many phages you find at the site of infection and elsewhere in the body and how well they kill the bacteria. We need to look at the ability of bacteria to be killed, biomarkers of dead bacteria. We need to see if patients improve clinically and how these factors relate to each other. We use mathematical modeling to understand how the numbers of phages in a wound correlate with the number of bacteria being killed. We then go back and look for better phages, and for things that work, we persist with. Things that don't work, we discard and proceed that way. And this is pretty much where we are now at IPATH in terms of our phage therapeutics program. Now, what about public health? There are also ways, we'll just close with this experience. You may have read about contaminated eye drops earlier this year. These were eye drops that didn't have any preservative in them. They are used by people who have the dry eye syndrome. If you have just allergies, you can put in eye drops that have benzalkonium in them. You only have to use them once or twice a day. But benzalkonium can't be used every 2 or 3 hours because it causes problem for the cornea. And so for people with dry eye, they market eye drops that don't have any preservative. And by not having preservative, bacteria can grow in the eye drops either from the manufacturing plant or being contaminated after they're out of the manufacturing plant. There was an outbreak of extensively drug-resistant Pseudomonas contamination in eye drops that came from 2 different manufacturers outside the U.S. And you can see that by the time of this report last spring, there had been 81 cases in 18 states. 14 patients had totally lost their vision. Some of them had to have one of their eyes removed. There have been a couple of deaths because these bacteria can spread to other parts of the body. And the organism that they had was a very, very bad one in terms of the resistance genes that it carried with them. This shows you what one of the corneal ulcers looks like when the Pseudomonas attacks the eye. And again, the eye drops that we have are not active against them. So David Pride and I got in touch with the CDC that have been investigating these and asked them if we could get some of these Pseudomonas isolates that have been isolated from these patients, and they sent us some. You can see here, if you look at the resistograms, these are 3 different organisms, PS 747, 748, and 749. And this is how much of each antibiotic is required to kill them. You can see that greater than is an indication that we don't even test at higher concentrations of antibiotics because they're toxic. You can see these are not very friendly bacteria that are coming out of these patients. David was able to go to a collection of phages that had come from the Department of Biology here called jumbo phages, and this is a grid that shows across the top different Pseudomonas isolates. Those I just showed you, 747, 48, and 49 are up the upper left. And then down the y-axis, these are different phages. One of the nice things about phages, you can name them anything you want. And you can see that people have great imagination in terms of some of the phage names. But 5KZ kills very well those 3 Pseudomonas isolates. Now, one of the nice things about outbreak phage use is that if you have an outbreak that is caused by a given organism that gets into a eye drop batch in India or anywhere else, and it is found in a patient in Michigan, if a patient in California is using the same eye drops, you know it's going to be the same organism. So you can prepare one set of phages and go after all the patients in that outbreak. So we've been, based on this experience, the CDC has put up on their website about the availability of phages, and we're looking for people to treat, but we're also working with CDC now to analyze the outbreaks that they're coming up with in their outbreak investigations branch so that we can work with them to create phage libraries for new outbreaks that come down the road. We're now in kind of IPath 2.0, and what we're doing from the research perspective is developing a kind of a multidisciplinary program that includes basic research, developing better phages, clinical research, treating people with with bacterial infections, and trying to learn from that what we need to do to improve the phages, and then feeding that back into the translational and basic science portion. And then we have Natasha Martin and others working on modeling to understand better the quantitative relationships between phages in vitro and in vivo. And then we're moving into expanding phage availability at UC San Diego in the hospital with phage units that will be available for people with infections that are amenable to phage therapy. So I'll close now and just say that antibiotics, to get back to kind of where we started, have one advantage of being very broad, but the disadvantage can also be they disrupt the microbiome in a major way and select for organisms that are even more resistant than what you were treating to start with. Bacteriophages have a narrow spectrum, and that can be a challenge if you're needing to find a phage for a particular organism. But if you already have a big library, that can be solved. And when you use a phage, you don't have to worry as much about disrupting the microbiome. It takes a long time to develop an antibiotic, years, hundreds of millions of dollars. We could go over to David Prod's lab today, and by next week have a handful of phages against any organism that you show us today. I'm a little hyperbole here, but the point is the phages are everywhere. You can think of us as just coral reefs that grow bacteria that phage attack. So we're just basically serving as phage discovery mechanisms, walking around with so many bacteria and phages in our GI tract. And finally, it's not that expensive to make a phage, and it's getting less and less expensive to purify them. There are a number of indications outside human medicine. This is a figure that Stephanie put together for our paper, and just to make the point that people are already beginning to think about using them. They're actually using them instead of tetracycline in chickens to reduce the amount of antibiotic in livestock. Veterinary usage, you can think about using it to disinfect water and so forth and so on. So there are a lot of other places where phages may well be used. So I'll stop there, and I'm happy to entertain any questions or comments anybody has. Thanks very much.
Anyone have any questions or comments?
Yeah. So how fast? So compared to like that first example of Patterson, how fast are phages discovered compared to then? Now that the process has been refined more.
So I'm sorry, how fast are they discovered?
Yeah.
Do you mean for a given patient, or do you mean when you say how fast? What do you mean by when you say how fast are they discovered?
Yeah, for a given patient or given bacteria.
Well, there are kind of two levels of discovery. For some organisms that we treat a lot, like Staphylococcus and Pseudomonas, we already have big libraries that are available to screen with. And so with that, you just take your library and you can find a phage in 2 or 3 days. If it's an unusual organism, you need to go out to the environment and look for phages in the environment. And you generally go to where the bacteria live, because that's where their phages are. And that can take weeks to months if it's not a very unusual organism. So what we're trying to do is to continue to build the library of phages for some of these less common organisms so we don't have to go out and search for them every time. The patients give them to, generally, if we treat people intravenously, which is what we've been mainly doing, they can be given by the patient at home. They don't turn a hair when they get them. And so we send them home with a week's worth of phages. They give themselves phages twice a day and administer them that way. They're quite stable under those conditions. Phages are also being given by aerosolized routes for cystic fibrosis, for example, and with pretty good success in terms of the antimicrobial activity. People have used oral phages to treat cholera, so that there's some interest in trying to figure out how to use them orally. One of the challenges with that is that most phages are fairly sensitive to gastric acid, and you have to work on ways to get the phages through the stomach to the duodenum. And then they can be used topically for wounds and burns. Surgeons like to splash phages onto prostheses when they're in the operating room, but they like to splash antibiotics and other things on them too. It's fine with me, but the blood supply gets them there better in my view. So, hi, yes.
So we have a question from the audience in the Zoom session. One of the audience said that, what an inspiring talk, and how long does it take to set up a phage therapy? What is the biggest obstacle in the creation of the therapy process?
Right now, the biggest obstacle is actually, there are two obstacles, one of which is sometimes very easy, and that is finding the right phage. And that can be done, as I said earlier, pretty quickly if you already have a library that covers it. You then have to take those phages and grow them up and purify them. And what we need very badly here at UC San Diego is a large phage production facility that we could use to make up phages in large quantities to give clinically at levels of purity that allow us to give the doses we give, a phage production facility. It's not hard to do. The same kinds of facilities are the ones we use to make interferon and a lot of the proteins that people get treated with. But the biggest, but that's a bottleneck because we have to find somebody who can make them.
Well, thank you very much for
Thank
coming.
you.
And I hope you enjoyed this series this quarter, and it's going to continue in the winter. So hope you can enroll.
you.
Thank you. Have a good evening.